for a postdoc in Tel Aviv University from 2001 and 2003, where she worked on dynamic force spectroscopy. And then she moved to US, first at the National Institute of Health, and then finally from 2007, she moved to UCSD, where she's still now. So, as far as I understand, in her career, she translated from a condensed matter background to biology. And right now, she applies theoretical physics methods to understand biological problems. And for example, she explained the organizational processes taking place in the nuclei of living cells, right? And today's talk is about these things. And the title is Specificity and Universality in Neuronal Communication. The floor is yours.
Thank you, Dario, for the introduction. Is the microphone working? Yes. It's a pleasure to be here. It's a pleasure to be in Brazil and to visit the Institute for Theoretical Physics. Thanks for inviting me. I would like to tell you about some of our recent research on synaptic transmission. Synaptic transmission is a process by which neurons communicate with each other, and it's a process which ultimately enables us to feel, to think, and to act. I find it surprising every time I think about it that our modern picture of the brain as consisting of individual cells is actually relatively recent. It's only about 100 years old. This is one of the beautiful drawings by Ramon y Cajal, which he made using Indian ink. This particular drawing depicts neurons from the part of the brain known as dentate gyrus. It's a region of the brain that is responsible, among other things, for our ability to navigate new environments. Note, for example, how this afferent fiber cell, labeled A, makes connections with the dendrites of the granule cell, and at the same time, the collaterals of the fiber make synaptic connections with the pyramidal basket cell. Ramon y Cajal drawings not only precisely reproduced neurons, but also showed that the brain is not a single continuous network, which was the dominating view for quite some time. Rather, the brain consists of discrete cells, nerve cells, neurons, and these cells are separated from each other by physical gaps. And this immediately raises the question, how do neurons communicate across these gaps, across synaptic gaps? And this is a central question of this talk. Here's a more modern visualization of neurons making synaptic connections. These neurons were isolated from a marine snail. Visualized in the center in green is a mechanosensory neuron, which is seen to form synaptic connections with two motor neurons. So before we talk about synaptic transmission, let's make quickly a step back and look at into the process that immediately precedes the transmission of information from one neuron to the next. And this is a process of propagation of information along a single neuron. Propagation of information along a single neuron is electric in nature. And thanks to Hodgkin and Huxley, we have a theory that describes the propagation of nerve impulse, also known as the action potential, along the terminal, along the axon of a single neuron. One of the things that makes Hodgkin-Huxley theory so powerful is that it connects the phenomenology of the action potential, the phenomenology of the nerve impulse, to the molecular details, in this case, the ion channels that are embedded in the membrane of the neuron. In contrast to the action potential, the electric nature of the action potential, the transmission of information from one neuron to the next is chemical in nature. Neurons use a chemical signal to communicate with each other, and the transmission of a signal across synaptic gaps is what is responsible for our perception, for movement, for our ability to learn. And therefore, the transmission of information has to be both ultra-fast and precise. And it is. The two relevant timescales are very short. The timescale that describes the duration of the release of the chemical signal is, in the synaptic gap, is less than a millisecond. And similarly, the timescale that describes the delay between the trigger and the initiation of the release of the chemical signal is less than a millisecond. Another remarkable feature of synaptic transmission is that the strength of synaptic connections is not fixed. It is plastic. It can be enhanced or diminished at pre-existing synapses. And this is in response to our experience, in response to sensory stimulation. And this is a phenomenon known as synaptic plasticity, which underlies memory and learning. And unlike the situation with the action potential propagating along a single cell, there has been no theory that would describe how information is transmitted from one neuron to the next, at least not a theory that, in the spirit of Hodgkin and Huxley, would connect the molecular machinery of the synapses to the phenomenology of synaptic transmission and even further to synaptic function. And this all has been more than enough of the motivation for us to try to make at least a first step and try to formulate a quantitative framework that would be both phenomenologically accurate and mechanistically realistic, and that would allow us to connect the molecular level to the phenomenology of synaptic transmission and perhaps even further to synaptic function. So since molecules are going to appear in our theory, let's briefly review some essential molecular details. So neurons communicate using a chemical signal. The chemical signal is in the form of neurotransmitter molecules. Neurotransmitter molecules are stored inside synaptic vesicles. Synaptic vesicles, in turn, are localized at the terminal of the presynaptic cell. Presynaptic means transmitting cell. Postsynaptic means receiving cell. So synaptic vesicles are localized at the terminal of the presynaptic cell. Now, how do neurotransmitters get out of the synaptic vesicle? This happens when the action potential, the spike, arrives at the terminal of the presynaptic cell. The resulting depolarization of the membrane causes calcium ions in the membrane to open, because these ions, calcium ion channels to open, because these ion channels are voltage gated. And as a result, calcium ions realize suddenly that they can now flow down the electrochemical gradient, which they do. And so concentration of calcium increases very rapidly and drastically within the terminal of the presynaptic cell. And the increase in calcium concentration causes synaptic vesicles to fuse with the membrane of the presynaptic cell. Fusion culminates in the release of the content of the vesicles, the neurotransmitters, into the synaptic gap, whereas they complete their mission as chemical messenger by binding to the postsynaptic neuron. The message has been transmitted. Here's the electromicrograph of a neuromuscular junction, in other words, synapse between nerve cell and muscle cell, where this is an electron microscopy image, which also visualizes synaptic vesicles at different stages of their fusion with the presynaptic membrane. Here, for example, is a vesicle that is docked at the membrane. Here are the vesicles that are halfway through their fusion process with the presynaptic membrane. And here's a vesicle that has fused with the membrane and presumably has released the content, the neurotransmitters, into the synaptic gap.
Sorry, the neurotransmitters, are they called back or they remain there and the presynaptic creates other, or how it works?
Are you asking about the replenishment of neurotransmitters?
Yeah, if they remain in the postsynaptic or they are called back?
They bind to the postsynaptic cell.
Okay.
And the receptors, ligand-gated receptors on the postsynaptic cell, as a result, open.
Okay.
Which causes depolarization of the membrane of the postsynaptic cell, right? And so the signal propagates further.
Right.
And it happens much, much faster than it took me to explain it.
So the presynaptic reproduce new neurotransmitters?
There are multiple, it's a very good question. There are multiple mechanisms by which the presynaptic cell replenishes its pool of vesicles. Yeah, so some of them can be recovered back. There are vesicles that are assembled from scratch, and there are multiple mechanisms. Yes. Other questions at this point? So even from this minimal description, it is clear that an essential step in synaptic transmission is membrane fusion. Fusion of the membrane of synaptic vesicles and the membrane of the presynaptic cell. However, membrane fusion is known to be inherently very slow process. Even if you put two lipid membranes together next to each other, they will not fuse on any reasonable time scale. There is a high kinetic barrier on the order of 100 kT associated with drastic structural rearrangements within the membranes at the membrane-water interface. So how is it possible then for synaptic transmission that has membrane fusion as a required step to occur on a millisecond timescale? The key to speed is a conformational transition of specialized proteins, which are called snares, snare proteins. Snare proteins tether synaptic vesicles to the membrane of the presynaptic cell. And when calcium concentration is increased within the terminal of the presynaptic cell following the action potential, snare proteins undergo a conformational transition. It's a zippering kind of conformational transition, during which they have no choice but to pull the two membranes to which they are attached together. And this speeds up their fusion, the fusion of the membranes, enormously by orders of magnitude. So in other words, the free energy released during the conformational transition of snare proteins, conformational transition triggered by the increase in calcium, the free energy released is used to do mechanical work to pull the two membranes together. The kinetics of synaptic transmission has been studied both in vivo and in vitro. In vivo experiments using real synapses, they use caged calcium to rapidly elevate calcium concentration throughout the terminal of the presynaptic cell, and they use electrophysiology to measure the response of the synapse to a stimulus. And among the many things that these experiments have revealed was that there are, in fact, two distinct pools of synaptic vesicles. One pool is called readily releasable pool, or RRP. Vesicles in the readily releasable pool are tethered, docked, and primed at the presynaptic membrane, and as the name of the pool suggests, they are ready to release the neurotransmitters once the critical number, let's call it N, of snare proteins undergo the conformational transition in response to the increase in calcium concentration. In contrast, vesicles in the reserve pool, they are not tethered to the membrane, and so they do not readily fuse. Rather, this pool supplies vesicles to the readily releasable pool from which they can fuse rapidly following the conformational transition of N snare proteins after the protein sense the increase in calcium concentration. There is separation of time scales here, very conveniently. The conformational transition of a snare protein once calcium concentration is increased is a fast step, while the transfer vesicles from the reserve pool to the readily releasable pool, related to your earlier question, is a slow time scale. Now, these are in vivo experiments. There are also in vitro experiments that try to reproduce at least some features of neurotransmitter release, vesicle fusion and release in synthetic synapses, synthetic vesicles in complex with reconstituted proteins, including reconstituted snare proteins. When calcium is injected in the system, so these experiments use advanced optical imaging in conjunction with electron cryotomography. And when calcium is injected into the system, these experiments, synthetic systems, are able to reproduce many essential features of vesicle fusion and release, including, crucially, the millisecond timescale. And so these experiments observed two distinct morphologies at the contact between the synaptic vesicles and whatever mimics the presynaptic membrane. The vesicles in the so-called point contact fuse readily once calcium is increased and N snare proteins undergo the conformational transitions. So these vesicles mimic the readily releasable pool in vivo, while vesicles in the so-called extended contact with the membrane do not fuse readily. Rather, they get stuck in a hemifusion, in a fusion intermediate known as hemifusion diaphragm, and escape from this intermediate step constitutes the slow step. The slow step. So. Question, yes.
Sorry, I'm not the biologist. In vitro means in a lab, but how does the nerve know if it's in a lab or in a living thing?
Say that again, please.
In vitro means it's in a lab, right?
everything is in the lab. In vivo means it's in an intact synapse, not quite inside a living animal. The synapse is extracted, but it's a intact synapse. While in vitro means, so people synthesize synaptic vesicles. They reconstitute the proteins and artificially make them, get inserted into the membrane of the vesicle. So in this sense, this is synthetic experiment. And so what was the question then?
So it's not molecules, it's something else? I'm sorry, I don't know what...
Well, one can synthesize lipid vesicles, for example, out of lipid molecules.
So they're trying to construct an artificial nerve, is that?
It's a little bit maybe not yet close to the nerve, but it's artificial synapse in a way. Yeah,
okay, thanks.
Artificial synapse. And so they use fluorescence signal to see when the membranes of the whatever mimics presynaptic vesicle and the postsynaptic presynaptic membrane fuse, and they see when the content flows between the vesicle and the presynaptic membrane. So in vivo and in vitro experiments, obviously they have very similar features, but there are also some differences. The interpretation of the states is different. Kinetic schemes look different. But to our delight, we realized that all these experiments, in vivo, in vitro, those in vivo experiments that use actually different approaches to measure the response of the synapse to stimulus, they all can be encompassed by a single unifying kinetic scheme. And in this unifying kinetic scheme, synaptic vesicle fusion can be thought of as a process that occurs along two coexisting pathways. These pathways can be thought of as originating in a fast and slow vesicle pool. Each of the pathways contains a fast step. And remember, in vivo, which also means in an organism, fast step is the calcium-triggered conformational transition of SNARE proteins. But in addition, one of the pathways contains a slow step. And in vivo, the slow step is the transfer of vesicles from the readily releasable pool to the, from the reserve pool to the readily releasable pool. The replenishment of the pool is a slow step. And so the fact that all these experiments can be captured through a single unifying scheme means that, at least in principle, one can try to write down a single unifying theory to describe synaptic transmission, as opposed to having, a theory for each individual synapse or each different type of experiment. And this is a kinetic scheme that forms the foundation for our quantitative framework. I will skip the details of the derivation and let me go straight to the results. One of the rewarding aspects of our quantitative framework is that it is completely analytically tractable, meaning that it allows us to obtain closed form analytical solutions for the quantities that people measure in their experiments. The equations, unlike what your first impression might be, are actually quite simple. They are very intuitive, in fact, intuitive enough so that we can restate them in words. For example, a quantity that is almost always reported in experiments that measure kinetics of synaptic transmission is the release rate, which is defined as the rate of change in the number of fused vesicles as a function of time, and at any calcium concentration of interest. And so our equation tells us that there is a very simple structure that determines the release rate. It is essentially the sum of the fusion time probability distributions along the fast pathway and the slow pathway, and each is multiplied by the size of the corresponding vesicle pool, the fast pool, readily releasable pool, and the reserve pool. Now, the release rate reaches a peak at some time t, and so the quantity, the peak release rate, is an essential characteristic of a synapse. It is simply the maximum of the release rate. And once again, the analytic equation tells us that what governs the peak release rate. In particular, it tells us that the calcium's dependence of the peak release rate enters primarily through this calcium-dependent fast rate constant, which, remember, is the rate constant that describes the conformational transition of SNARE complex. Now, if we have the release rate, we can, of course, calculate the cumulative release, which is simply the number of vesicles fused by time t. And once again, the structure of the solution is very simple. So in order to make these equations practical, meaning directly applicable to experimental data, we need to know an explicit functional form for the calcium dependence of the fast rate constant. It enters the peak release rate. It also enters both release rate and cumulative release through these probability distributions. To determine the calcium dependence of the fast rate constant, the one that describes the conformational transition of snare complexes, we propose the following. We can think about a conformational transition of snare protein complex in its complex with the membrane as activated rate process, a thermal escape over activation free energy barrier along an emergent collective coordinate. And you probably recognize the classical Kramer's picture of diffusive barrier crossing. In our case, the height of the barrier is modulated by an external force. What is the external force? The external force is the chemical potential of calcium ions. Okay? And so with this picture, we can derive an analytic solution for the calcium dependence of the fast rate constant, the constant that describes the conformational transition of snare proteins. And this analytic solution tells us how the snare conformational rate is governed by the change in the barrier height. The change in the barrier height due to the change in calcium concentration following the action potential. And finally, perhaps the most curious result of this framework is this. So if we take peak release rate and non-dimensionalize it, divide it out by synapse-specific parameters, then the theory predicts this very simple-looking, at least, relationship between the non-dimensionalized peak release and non-dimensionalized calcium level. And this scaling is predicted to hold for all synapses. In other words, the data from all synapses should somehow collapse on this single master curve, which is somewhat intriguing because synapses are so very different in terms of their size, their particular variant of the molecule that serves the calcium sensor, in terms of their role in a functional role in the nervous system, and so on. But nevertheless, this is a prediction. So before we see how the theory is applied to experiments, I would like to say a few words about the value of analytical approaches. The stick in the sand approach is a reference to the story, probably apocryphal, of Archimedes, who used a stick to derive formula in the sand when soldiers invaded his house, and it didn't end well for him. But we are at the Institute for Theoretical Physics, so I assume that I don't need to work very hard to convince you about the value of analytic approaches. So a theory that is sufficiently general will apply not just at a particular set of experimental parameters, but across a broad range of parameters, so that when you get data from an experiment under different conditions, you don't need to derive a brand new equation to fit the data. Furthermore, a theory that is sufficiently general will apply not just to a particular system, but across different systems, in our case, across different synapses, whether this is a synapse that allows us to localize sound or synapse that allows us to contract our muscles, right? The same equation should hold. Now, the theory that is analytically tractable, first of all, I think that analytic theories are often useful guides for our intuition. But in addition, if you have analytic solutions for something that people can actually experimentally measure, then these solutions can serve as very convenient tools for extracting microscopic parameters from the data, because you can simply least square fit the experimental data. So in our case, from fitting the analytic expression that I showed you to experimental data on synaptic transmission, one can extract the following parameters. First of all, the kinetic and energetic parameters of snare protein complexes. What are these parameters? The height of the free energy barrier, the rate constant, and the sensitivity, the occupancy of the calcium sensor, which is simply the number of calcium ions that need to be bound to the calcium sensor for the snare protein complex to attain the transition state at any calcium concentration of interest. Question? Yes.
There's a danger I see, which is you're trying to apply this for all synapses. There must be some error bars, right?
Definitely, yeah.
Error bars must depend on the type of synapse you're trying to describe, right? So in some cases, the analytic solution might be perfect. In other cases, it might be very bad.
Of course.
Do you have an idea about when it's good, when it's bad?
I think we will probably see when we will actually apply the theory to data. Yeah.
Okay, but you don't know yet from the theory when it should be good and when it should be bad. You don't have an approximation that says this is valid in some limit in which I understand the parameters in which that limit is good.
The theory is valid. Obviously, there is a finite range of validity, but it is supposed to be valid quite broadly. There's no particular limit that would not be, you know, the theory works for a very broad range of calcium concentrations, for example, right? Okay. But you're raising, of course, a very important point, and perhaps when we see the actual application to the data, you will notice perhaps some scatter or some deviation from the curve, right? But one could, of course, be even more quantitative and find, for example, the expected variance, right, for certain quantities, definitely, yes. And since you asked this question, for some time, I really resisted the idea of, you know, really putting an effort in deriving a theory of synaptic transmission, even though, you know, by then we already worked on something else, and we realized that those approaches can work here. And the reason I resisted is because I just couldn't imagine that it is humanly possible to measure accurately enough events that occur on a sub-millisecond timescale, accurately enough so that a comparison with a relatively sophisticated theory would be meaningful. And I think that I was wrong. So some of the experiments that can be extracted from the feed of the theory to the data are these parameters of that describe the snare conformational snare complex, protein complex, the sizes of the vesicle pools, how many vesicles are there in the readily releasable pool and a reserve pool. As far as I know, there's no reliable method to extract these numbers, and the numbers vary dramatically from just a few for very small synapses to thousands of vesicles in a readily releasable pool. And finally, the theory allows one to extract this N, the number of snare proteins per vesicle. How many proteins per vesicle do you need to be able to achieve this highly synchronous fast fusion? 1, 10, 100, So let's now see how this so far abstract theory performs when it is confronted with reality. And here is a piece of reality. In fact, 16 different pieces of reality put on the same plot. What is measured here is peak release rate as a function of calcium concentration. If you remember, peak release rate is one of the quantities for which we have an analytic expression. And different symbols correspond to different synapses. So there are 16 different data sets from 16 different papers. As far as I know, this is everything that exists in the literature. Yes?
In vitro is just those 2 points over there? In vitro?
This is what people measure. Yeah.
Yeah. Okay, okay.
Yeah. But there are very rich data sets here for in vivo systems. Yeah. In vitro experiments are very challenging.
Curious. Okay.
So this list of synapses is really quite diverse. It's quite rich. It contains, for example, the synapse called the calyx of Held at different stages of development. Calyx of Held is a relatively giant synapse of about 20 microns, 20 micrometers in diameter from the auditory central nervous system. The calyx of Held synapse is responsible for our ability to localize sound, to be able to estimate where the barking dog is or where the car is approaching, from where the car is approaching. And the way that we localize sound, at least for low-frequency sound, is by calculating the difference in the arrival time of sound between our two ears. And remarkably, humans can detect the difference in the arrival time of sound between the two ears of just a few microseconds. But there are species that can do even better. For example, the barn owl. Sorry, I think I maybe went ahead of myself. Yeah. For example, the barn owl, which can detect the difference in the arrival time of sound of just 1 microsecond. And this allows it to detect a mouse covered under deep snow. It cannot see it, and any other bird, none of the other birds can see it, but it can hear it. this is a little story of the calculus of Held. This list also contains a tiny synapse, parallel fiber molecular layer in the neuron. It's a synapse of only 1 micron in diameter from the cerebellum. The list also contains inner hair cell synapse, which is responsible for converting sound waves into the electric excitations of your auditory nerve, thereby converting the perception of sound into the sensation of hearing. There's also retina bipolar cell synapse and so on. And so we have an equation for this quantity, peak release rate. And so we can fit each of the data set with this equation. And once again, we don't need 16 different equations, right? It's one equation that is used to fit each data set. It is worth pausing for a moment and appreciating the dependence of the release rate on the force that drives the release. The force that drives the release is the chemical potential, comes from the chemical potential of calcium ions. And we can see that there is exponential dependence of the release on the force. And this exponential dependence explains quantitatively the remarkable synchrony of the vesicle fusion and release. Because exponential sensitivity means that once calcium concentration is increased, the synaptic fusion machinery turns on very rapidly, exponentially rapidly. And similarly, once calcium is depleted, the synaptic fusion machinery shuts down also very rapidly. From the fit, we can extract various parameters, like I described a few moments ago. Here, I only show the values for 2 such parameters. The values for the parameters, for the activation free energy barrier at resting value of calcium concentration in the units of KT. So we have protein folding people. You can see that the resulting values are very typical, between 10 and 35 perhaps, for the activation energy for protein conformational transition. And here are the values for the calcium sensor occupancy, meaning the number of calcium ions that need to be bound to the molecules that act as calcium sensor for the molecule to achieve the transition state on the way to the conformational transition. And most of these numbers are between 1 and 5, but some of these numbers really puzzled us because for several synapses, the number is actually greater than 5. And how can they possibly be greater than 5 if there are only 5 binding sites in a calcium sensor, five calcium binding sites? So we, of course, checked many times for all possible errors until we had a confidence to trust what the theory is telling us. And we realized that what the theory is possibly telling us is that some of these snare proteins are actually assemblies of multiple snare complexes working as a single unit. And therefore, you have more than 5 binding sites for calcium. And the existence of such snare superassemblies has already been hypothesized by researchers in a study using a very different approach, using electron microscopy, where they observed a mysterious protein, mysterious symmetry in protein density.
Now, you said that you also were going to measure how many snare proteins per vesicle. Yes. So does that number correlate with this, like that you have more snare proteins in these synapses that have a larger number of calcium?
It's a very good question, indeed. But this goes back to the quality of the experimental data. I shouldn't complain about the quality. I think this experiment is insanely complicated, right? So quality in terms of we definitely don't want to face the danger of overfitting. And so in this case, we did not use N as a free parameter. But in a moment, I'll show you an example where the data set was so extensive where it was possible to identify, to extract N. But then you are also asking an interesting question about correlations, right? Yeah, so when there will be more data, we will be in a position to answer. Yes. Here are some more examples of comparison between theory and experiment. This is a very rich data set for another quantity, cumulative release, for which we also have an equation as a function of time for in this case, for calculus of Held, measured at different values of calcium concentration. And so the data set is rich enough and high quality enough for us to do a global fit of the corresponding equation for the cumulative release, corresponding solution. Global fit means one equation applied across all these different curves, all the different calcium concentrations. And in particular, it allows us to try to answer the question of N, how many snare proteins per vesicle is needed for fast synchronous vesicle fusion and release. Though the theory makes no assumption about a particular value of N, it works for any N. However, it tells us that there is something special about N equal to 2. So when N is equal to 2, we can see from this graph that then the release rate, first of all, it avoids this dangerous instability, very high release rate on short time scales, but at the same time, it achieves the peak sooner than any other N, right? So there's a nice balance between stability and precision. Nevertheless, without any assumptions, we do a global fit to this experimental data, and actually the best fit does occur with N equal to 2. And I'm very curious to see what N comes out of the data from other synapses when the data are ready, and it is quite possible that N will be different for those synapses. So this is the number of snare proteins per vesicle. Here's more comparison between theory and experiments. These are synthetic systems, and once again, we can feed the data, extract the parameters. Here's a very interesting data set. It actually is from one of the pioneering papers from Chuck Stevens' lab, one of the pioneers of synaptic transmission. They studied experimentally synaptic transmission in frog neuromuscular junction, which is the synapse between nerve and muscle in a frog. And on this figure, the data points are their experimental data. And the dashed curve is something that these authors used to try to explain the experimental data, because this was, I guess still is, considered the golden standard in this part of neuroscience, the so-called force power model. And the force power model postulates that the release rate scales with calcium concentration as concentration to the power of 4. For comparison, the solid line is our theory, which clearly performs much better than the force power model. Everything I told you so far in terms of comparison between theory and experiment was about how we can use the analytic form of the solutions to feed the data and to extract microscopic parameters that are unique for each synapse, that uniquely identify each synapse, right, that are specific for each synapse. And now I'd like us to look at not what's specific, but what's universal. So here's, again, the plot for the peak release as a function of calcium for 16 different synaptic preparations. And I think that this plot is a great illustration of why that's so challenging for a theorist to try to think about synaptic transmission in unifying terms. Well, because synapses are so very different. On the one extreme, there are synapses that run along the path from our ear to the brain, and these synapses are responsible for preserving sub-microsecond timescale events. On the other extreme, there are synapses that control release of hormones, such as insulin. These synapses operate on the timescale of minutes. And so when you cover 3 orders of magnitude in calcium concentration and consider all 16 systems, you get access to 10 orders of magnitude in the peak release rate, not factor of 10, 10 orders of magnitude. This is a striking dynamic range. And nevertheless, the theory predicts that once you non-dimensionalize peak release and calcium level by the synapse-specific parameters, then all the peculiarities of the individual synapses should no longer matter, and all these data should somehow obey this universal curve. And so this is what we did. Upon non-dimensionalization of peak release and calcium level, this is what happened to the data. Okay, so it works quite nicely, and this tells us that despite the striking diversity of synapses in terms of their size, in terms of their sensitivity to calcium, in terms of the particular molecular isoform of their calcium sensor and so on, in terms of their role in the nervous system, synaptic transmission obeys a simple unifying universal scaling law. And this is confirmed by the collapse of the data on the curve. And this is where I think it's interesting to think about the question you asked earlier about, for example, why there are outliers and what's special about the synapses. And is it a problem with experiment, or is it something inherently different about the synapses? But I think the collapses work so well that we can actually afford picking at the individual points. Yeah.
Yeah, just make sure I understand. The C there is rescaled calcium concentration.
Not calcium level, yeah.
Calcium
so,
level.
Calcium always enters with logarithm because it ultimately comes from the chemical potential. But yeah, rescaled calcium, rescaled trigger, right? And this is the rescaled peak release.
And what kind of factor are you using to rescale?
What kind of what?
Factor, like you rescale by what, for instance?
We just divide out by synapse-specific parameters.
All right.
So peak release, you can see that peak release versus calcium is very different for different synapses, right? But once you divide a peak release by a combination of parameters that are unique for a given synapse, and you do the same for calcium level, then you get this equation, which is there's no longer anything specific about individual synapses, right? And it works.
Okay.
So now that we have some quantitative framework with which we can describe synaptic transmission quantitatively, and we see that the framework is in detailed agreement with a wide range of experimental data, we can start thinking about what ultimately matters most for biology, namely biological function. After all, biological function is what sets life apart from non-living matter. And biological function of synapses, in particular, is their role in memory and learning. And so hopefully we now have a quantitative framework to address questions of memory and learning in a quantitative form. A remarkable feature of human nervous system is that it is functional at birth. Human babies can see, they can hear, they can breathe, they can suckle, but the capabilities of human infants are actually quite rudimentary compared to those of some other species. For example, many birds can fly shortly after they hatch from the egg. Here's barnacle gosling. It just hatched, and it has to dive over 400 feet from the nest on the top of the cliff, at the top of the cliff, to reach its parents, who are hanging out on the grass below. And remarkably, it managed to arrive safely all in one piece. Maybe even a more dramatic illustration is this wildebeest calf. It has just been born, literally, seconds ago, and it's already trying to stand up. It may take a few attempts. But within 2 minutes after birth, it is standing and is about to run. So these are just some striking illustrations of the fact that that nervous system is functional at birth, and that connection between neurons are set during development, before birth. But this then raises a very interesting and I think rather fundamental question. If our nervous system is wired so precisely, if connections between neurons are set during development, then how is it possible that we can memorize, that we can learn? How is it possible that we can modify our behavior, right? So the reason that this is possible is that nervous system is plastic in response to experience. the embryonic connectivity of nervous system is only a draft of the neural circuits that exist in us when we are adults. Synaptic connections can be formed. Their strength can be enhanced or diminished in response to sensory stimulation, in response to our experience. This is a phenomenon of synaptic plasticity. So let's look into what plasticity means at the level of a single synapse. Some synapses, in response to repeated stimulation, first exhibit a strong response, but the response decreases, fatigues rapidly with continuous stimulation. In contrast, other synapses first respond very weakly, but the response increases with sustained stimulation. So the first scenario is known as short-term depression, and the second scenario is short-term facilitation. In fact, these two examples come from the same creature, crayfish, which has in its abdomen two groups of muscles, phasic muscles and tonic muscles. Phasic muscles are responsible for fast, powerful contractions, which crayfish uses in swimming and also in escape response, for example, as here when it is attacked by a triggerfish. While the tonic muscles is a superficial layer of muscles that produce finely graded sustained contractions. And these muscles are used in postural control. And so it turns out that these two groups of muscles, phasic and tonic, fast and slow, are innervated by neuromuscular junction with distinct forms of plasticity, depression and facilitation. Furthermore, it turned out that this is a general strategy of muscle control, not just in crayfish, but also in mammals, such as ourselves. Fast and slow muscles are controlled by motor neurons with distinct forms of plasticity, depression and facilitation. So the simplest way of studying the synaptic plasticity at this level of a single synapse is to do the following. Imagine you stimulate a synapse with a pair of stimuli, pair of action potentials, each of duration t, and these two action potential spikes are separated by time interval tau interpulse time interval. And you measure the response of the postsynaptic cell to this pair of stimuli, and let's say the amplitude of the first response is A1, the amplitude of the second response is A2, and so if the amplitude of the second response to the second spike is greater than the one to the first spike, then this is an example of facilitation. If the second response is less in amplitude than the first one, it's depression. And so we can construct the ratio of A2 over A1. The ratio is known as a paired pulse ratio. And study this ratio as a function of this parameter, tau, and the interpulse interval, the temporal window between the two spikes. When the ratio of A2 over A1 is greater than 1, that's facilitation, right? And interestingly, in many synapses, facilitation can increase the response to the subsequent spikes by more than twofold. Now, what is the mechanism of short-term facilitation for short-term depression? It is fairly easy to understand short-term depression. It has already been mentioned at the very beginning. All synapses naturally get depleted of the pool of synaptic vesicles. Vesicles are replenished, but on a shorter time scale, right? So in this sense, all synapses are naturally predisposed to synaptic depression. But apparently, some synapses have also facilitation that is so strong that it more than compensate for that intrinsic tendency for depression. And so what is the mechanism for facilitation? And this is something that, interestingly, is not really understood yet, which is quite surprising given that short-term facilitation is thought to underlie working memory. Working memory is a type of memory that can be maintained over a short period of time, seconds, let's say, on the minute, and then is rapidly forgotten. Like those passwords that we can remember only until we enter them. There were a number of hypotheses of short-term facilitation that were proposed to explain it, and now that we have a quantitative way of thinking about synaptic transmission, we are in a position to test this competing hypothesis quantitatively. So a classical hypothesis, which I think is beautifully minimalist, was proposed actually by some of the pioneers of the studies of synaptic transmission. It is known as the residual calcium hypothesis, and the idea is very simple. The idea is, as a result of the first stimulus, spike, calcium enters into the system, binds to calcium sensors of SNAP proteins. The proteins do their thing, vesicle fuse, right? But some of the calcium ions linger in the terminal, presynaptic terminal. And so when the second spike arrives and there is a corresponding wave influx of calcium, this new calcium is added to the pre-existing residual calcium. So the calcium level during the second spike is higher as a result. And because the calcium concentration is higher, the release rate is faster, and the postsynaptic response, the response postsynaptic response is higher. So that, I think, makes complete sense, right? Now we can quantitatively assess this hypothesis by estimating the change in synaptic strengths due to the residual calcium using our equations. We can estimate the change as the ratio of the cumulative release. We have an equation for cumulative release for the final spike and the initial spike. And the resulting ratio can be approximated in this very simple form. This is a calcium-dependent rate, a constant of snare conformational transition, for which we have analytic solution, right? And so to evaluate this expression, all we need is the actual values for the calcium concentration, that leftover calcium, residual calcium, and calcium concentration during the action potential. And people have measured these concentrations, which is very challenging for various reasons. So the calcium concentration during the action potential is in the range of 25 and 100 micromolar, while the concentration of residual calcium is in the tens or hundreds of nanomolar. So several orders of magnitude apart. And so evaluating the rate constants at these values of calcium concentration, we find that the residual calcium under the most favorable conditions, favorable in terms of supporting the residual calcium hypothesis, we find that the change in synaptic strengths can indeed be greater than 1, which means that indeed it can lead to facilitation, but it is only marginally greater than 1. And therefore, it cannot explain the data on many synapses where facilitation increases the response by more than a factor of 2. And so this justifies us to consider a more complicated scenario, and there were quite a few hypotheses that have been proposed, and I'm looking at the time, so let me just give one quick example of this alternative scenario. This is the so-called facilitation sensor hypothesis, which in a way is a modification of the residual calcium hypothesis. And the idea is this. So here's a presynaptic vesicle. This is a presynaptic membrane. This is calcium channel. Here's the main calcium sensor that binds calcium and makes snare proteins undergo the conformational transition, right? But in addition, the hypothesis says there is a facilitation sensor, which can be thought of as another variant of this molecule. And in fact, people did genetic mutation experiments trying to knock down this variant, and they indeed saw that facilitation goes away. So there's a serious support of this scenario. So we can once again estimate the change in synaptic strengths as a ratio of the cumulative release for the final and initial spike, right? And we can easily incorporate this scenario into our theory using just a handful of parameters. One parameter is the time scale on which residual calcium is removed from the system. There are mechanisms that do this. Another parameter is the time scale that describes replenishment of the readily releasable pool. And finally, the parameters that describe the interaction between facilitation sensor and the main sensor. And by the way, the idea is that the facilitation sensor has slow kinetics and high calcium affinity. Slow binding kinetics means that it binds calcium with a delay, so it only binds calcium and gets activated during the 2 spikes, right? after the first spike is gone. But high calcium affinity means that once it binds calcium, it holds on to it strongly. And so once it gets activated, this facilitation sensor interacts through an unknown mechanism with the main sensor and amplifies the action of the main sensor, which is why what leads to facilitation. Anyway, so by incorporating this scenario, we arrive at the following very simple expression that's supposed to describe the change in synaptic strength, or the paired pulse ratio that we defined minutes ago. And this single equation is supposed to describe either depression or facilitation for a given synapse, depending on the interplay of this time scale and how they also compare to the time scale that describe the temporal window between the two spikes. Fortunately, there are experimental data in the literature, which we were able to find, that measure precisely this quantity, the paired pulse ratio, by varying the interpulse interval for several very different synapses. So the experimental data, they come from different groups. The experimental data with the error bars, usually a good sign, are symbols. And the curves are all generated by this one single equation. From the feet, we can extract these three parameters, the two time scale and the interaction constant. And we can see that, for example, retinal ribbon synapse is a depressing synapse because the paired pulse ratio is always less than 1, but the degree of depression decreases as we increase the time between the spikes, while all the other synapses in this collection are facilitating synapses, right? The ratio is greater than 1. I'm a little worried about the time, so I wonder if we should wait until the end. Thank you. So if we stare a little bit at these numbers, it begins to make sense why some synapses are facilitating, other are depressing. It's basically the competition of these time scales. So I think we really have run out of time, I'm not trying to start a new talk. I just wanted to point out one last thing. So this is a cartoon of an enveloped virus about to infect a cell. Enveloped virus is a virus that is covered by a lipid membrane. HIV is an example. Influenza is another example. Coronavirus is also a membrane virus. And so since the virus is covered by membrane, in order to inject its genome into the cell, it has to fuse with the cell. And so some time ago, with my former student, Yaojin Jiang, we wrote down a statistical mechanical theory to describe the invasion strategy of enveloped viruses. And in particular, we arrived at the following scaling relationship between the rate of viral fusion and release of the genome and the trigger. In the case of influenza virus, the trigger is the drop in pH inside the endosome. Now, what I told you today about is something very different in many ways, right? Something, it's a synaptic transmission that occurs on a very different length scale, very different time scale, but some of the equations I showed you today look very similar. In fact, the scaling looks just the same. And perhaps by now it's not that mysterious anymore, because what these two very different in biological sense processes have in common is they face the same problem. Both systems face the same problem. In order to accomplish whatever they want to do, infect a cell or transmit a signal to another neuron, they have to overcome this insurmountable 100 kT activation energy barrier. And to me, what's striking is that both systems have discovered the same solution to this problem. The solution is to recruit fusogenic proteins, specialized proteins. The protein sends the trigger, and as a result of that sensing the trigger, the proteins undergo conformational transition, during which they release free energy that is used to do mechanical work to pull the two membranes together. As a result, the resulting potential has a very modest barrier, and so the time scale for fusion is reduced by orders of magnitude. And what I find striking is that the systems are obviously very different. The fusion proteins are very different. In the case of influenza, the protein is hemagglutinin, for protein folding people here. For now, the trigger is very different. The synaptic transmission, the trigger is calcium. In the case of influenza fusion, the trigger is low drop in pH. But apparently, quantitatively, the solution that the systems have discovered is the same. Furthermore, there are many examples in biology where important things must happen, must proceed through membrane fusion. Fertilization of a cell, by the sperm is only possible by thanks to fusion between the two cells, right? The way that our muscles grow is actually by fusing of many cells. So our muscle fibers are actually giant single multinucleated cells. There are many other examples. And so there are strong indications that all these different systems that face the same problem, in order to do what they want to do, they have to overcome, to beat the speed limit of membrane fusion. It looks like they might also have discovered the same solution quantitatively. Let me quickly summarize. I shared with you a quantitative framework that was intended as a first step toward a theory to bridge the molecular description of synapses to the phenomenology of synaptic transmission and begin to answer some questions about synaptic function, memory, and learning. The theory is shown to be in detailed quantitative agreement with experimental data on a wide range of synapses. The theory is analytically tractable, and so one advantage of that is that it allows one to extract easily microscopic parameters that uniquely identify a given synapse. And complementing this specificity is the universality. Despite the striking diversity of synapses and their properties and their role in nervous system, synaptic transmission is shown to obey a simple universal scaling law, which we saw is confirmed through the collapse of the data on a master curve. The theory allowed us to try to address some questions about synaptic plasticity, and in particular, it allows us to now revisit the competing hypotheses of plasticity and to evaluate them quantitatively, to test them quantitatively. There were a couple of things I didn't have time to tell you about, and finally, we saw that there are very substantial hints that the same physical principles operate across the systems that traditionally are studied. They're traditionally labeled as belonging to different subfields of biology. And yet we can think of them in unifying terms, literally using the same equations. And this is my last and most important slide. I would like to thank my current and former group members for the pleasure of working together on various aspects of the physics of life. The earlier work on the viral invasion strategy that led us think about synaptic transmission was done together with my former graduate student, Yaojun Jiang. She is now an assistant professor in the Department of Physics at Johns Hopkins University. The theory of synaptic transmission I shared with you today was done together with the physics graduate student, Bin Wang. And I would like to thank our sponsors, National Science Foundation and the Simons Foundation, and thank you all very much for listening and for your questions.
Thank you very much. Time for more questions? Okay, we have a plethora.
Thank you. Very, very nice talk. Regarding this last statement about these commonalities of apparently very different processes, I was wondering about enzyme working, because you also need to, enzymes, the way in which they facilitate reactions is by lowering these activation barriers. And I'm wondering if it could be described within your framework.
So
what
in fact,
enzymes do, which is, it's not fusion, it's not exactly fusion of membranes, but it's the thing of lowering the barrier.
Yeah, but in this case, the fusogenic proteins are in fact a catalyst, right? This is what plays the role of catalyst to accelerate reaction, just like enzymes do.
So in the first case, before you talked about the virus infection, time scales and length scales, they are quite small. So I believe that a spatial theory is not needed to understand, a theory that includes diffusion equations or something like that. But in the virus case, is this relevant? Because things are larger, the size of the cell, and
what's the question about the viral case?
Is a spatial theory needed, a theory that takes into account the positions and the concentrations in space?
No, the equations are literally the same. I mean, obviously, the variables have very different meaning, right? But I showed you two equations, and you can interpret, drop in pH as a trigger, and change in logarithmic calcium as a trigger, and then you end up with the same scaling, right? So you do not need to introduce spatial dimension. You can really think about it in terms of activated rate process, escape over the barrier, where the barrier is modulated by an external trigger. Trigger is different in different things, right? And so because this conformational transition of snare proteins is the crucial step, is a rate-limited step, right? Everything basically is waiting for that step to happen. But the fusion overall is not merely a transition of a single protein complex, right? So there is a combinatorial factor in the equation that basically tells you about how a given system recruits n neighboring fusogenic proteins to do what they're supposed to do.
More questions?
When you introduced the facilitation sensor, there were some extra parameters that came with it, right? Some different time scale.
Three parameters. So.
And how do you set them? Do you set them independently by some other?
So the three parameters that I introduced in that scenario are the one parameter is the time. I know I went a little bit fast over that part. So one time scale is a time scale on which residual calcium is removed from the terminal, right? And so the faster you remove calcium, the less favorable the conditions are for facilitation, because facilitation wants that calcium, right? Now, the second time scale is a time scale on which the vesicles are replenished in the pool, right? And so the faster this happens, the better it is for facilitation, right? And the third parameter was what we call sigma, is the parameters that describe the interaction between the two sensors, the main sensor and this new facilitation sensor. And the mechanism is unknown. We can think about some ideas of what experiment one can do to get some insights into what the mechanism could be, but that was the third parameter. And then the fourth parameter was not really a free parameter. It was the temporal distance between the two spikes, stimulating spikes, but that is something that is controlled in the experiment, right? It's not something we have freedom, over which we have freedom.
Because before you had calcium in that expression, and then when you put the new parameters, calcium was gone. I guess you absorbed it. Oh,
calcium is never gone. Everything is
okay, but you didn't write it explicitly. I mean, in your expression, you absorbed it in some of your parameters.
Yeah. So calcium enters through the fast rate constant, which is explicitly calcium dependent, right? And that is the main dominant dependence on calcium in the whole story.
Hi. Very nice talks. Fantastic again. You haven't commented anything about the synaptic vesicles. So I imagine that they play a role in terms of size, composition. How could you comment on their variability? Eventually, they come up like as effective number that is going to be cooked up when you renormalize everything. But could you comment on? Yeah. They probably change in terms of specificities depending on the requirements.
Yeah, as you know, of course, the power of theoretical physics is that we don't need, for each of the component of the system, each vesicle, each molecule, we don't need the corresponding number of tunable parameters, right? That is a great thing. And so, in response to your specific questions about, the heterogeneity in the vesicles in terms of size and other properties. So this is all packed into the activation free energy barrier and the rate constant and the calcium occupancy, which is the language of energy landscape, is the width of the barrier. Because when we talk about the rate of conformational transition of snares, these are not really isolated proteins. These are proteins that are stuck in their membranes, right? So the conformational transitions of snare proteins in their complex with the membranes. And so I imagine that the size and the properties of the lipid molecules of the vesicles, they all are accounted for in those parameters of the free energy barrier.
Hi, I'm also going to ask about the free energy barriers. So do you require as assumption that this free energy released by this conformational change is all transduced into mechanical work? Like, couldn't part of it be dissipated and not be used for doing this work of fusing the membrane?
You know, it's a good question, but no, we make no assumptions about that. We just, so maybe a little bit more detailed picture. For example, the way it works with hemagglutinin, the viral proteins, influenza viral proteins, is that the conformational transition, it can be thought of as a two-step transition. One step is overcoming the barrier, and the second step is downhill. And so the energy, free energy release from the downhill stage is what is used to do mechanical work, right? And the barrier for protein conformational transition in the absence of the trigger, I displayed a table, right? The values were pretty high, you know, up to 35 kT, which is a large barrier for something that needs to be accomplished on a millisecond time scale. But with the trigger, the barrier goes down to just a couple of kT, which is nothing, right? But just thermal kick can you know, be enough for the protein to accomplish that transition.
More questions? Okay, perhaps I can ask a question that might be silly because it's based on a slogan that I vaguely remember. So it's based, it's about the facilitation and depression mechanism that you described. Now, if I remember correctly, certain psychological conditions like depression, anxiety, and so on, they are largely unknown, but one story about is that the neurotransmission is compromised, and there are, so you, with your theory, say something about drugs for that, or about the mechanisms, or about this stuff?
So, you know, when you have solutions in analytic form, you have in front of you all the parameters and their combination that makes up a given quantity, right? And I think this gives you a way to try to predict the effect of a certain drug.
Yeah.
Right?
Have you ever thought about applying in that particular?
I was asked about this, but I haven't, you know, put a lot of thought in that particular question. But definitely, you know, you have an expression for the rate of fusion and release as a function of those parameters. You have very concrete expression, right? And so, for example, you can look at this expression and see that if you lower the activation energy barrier by a certain amount, you know precisely how the rate will be affected. And then maybe your friend, molecular biologist, knows what kind of mutation to introduce into the protein to achieve the change in the barrier height. So in that sense, I can see that it can be quite, it can have that very applied value ultimately.
Okay, if there are no, we have last one.
Hey, so you said that some very different topics of biology were kind of connected through the same equations. So this kind of creates some analogy between them, right? You could think, oh, okay, this thing would be like. Have we learned something we didn't know about some area through this analogy? Because like maybe we knew another area and they are correlated, I don't know.
Yeah, it's an interesting question. There are many levels at which this question can be answered. So importantly, this is definitely not just an analogy, right? So the corresponding descriptions are basically identical, right? So it's more than just an analogy. In terms of whether we learned about one process by thinking about something totally unrelated, yeah, I think I briefly mentioned that with June, when she was a student in my lab, we were thinking about the way that viruses can infect cells, even though it seems impossible at first because it requires membrane fusion, right? And nevertheless, viruses, influenza can infect the cell on the time scale of minutes. How is it possible? And so we I think, understood something about how this is possible, understood in terms of equations, right? And then from reading and talking to people, I learned that something similar, analogous, using the word used, occurs in synaptic transmission, where the fusion occurs between synaptic vesicles and the membrane. And so there has to be the same problem, right? But except that now this fusion has to occur not on a minute time scale like for viruses, but on a less than millisecond time scale, right? And then I learned that apparently synapses use similar biological strategy. They employ those fusogenic proteins that sense a trigger, right? And do the work to bend the membrane and to induce fusion. And then the rest is just trying to translate it into the theoretical physics, right? And looks like it actually worked, which is stunning to me. But I think we were really fortunate that the neuroscientists who study this, certainly on the experimental side, the level of experiments is very, very high. So I think this data, of course there could be more and better, but from what they already can measure is just incredible to me. And so in this sense, we were fortunate to be able to apply the theory to concrete data, not just to simulated data, right? But to something from, real world.
Thanks.
Okay. Let's thank our speaker again. Yeah.